So good afternoon, and thank you. I would like to thank the organizer for first for all the time that they dedicate to the bioinformatics community, and I think that they are doing a wonderful job, and this is really great for all of us. And also for allowing me to present my work. So the title changed a little bit compared to what is in the program. And this is because, so in the program, I wanted to just tell about what I did during my PhD. But then I thought that some people know my work, the work that I did during my PhD. And so perhaps it would be interesting for them, and perhaps also for you, to know what I've started doing in New York, in Columbia University. So I've started very recently, so it will be a brief overview, but still. So the common point between the two projects is the fact that it's on unconventional microbes with complex genome architecture. And during my presentation, I will highlight the challenges of working on omics data with such unconventional species. So, first, I will start very basically by giving you the general context of my research. So as you know, of course, the genetic information is specified by the DNA constituting the genome. And bioinformatic analysis can predict functional regions of genomes, such as genes, for example. But sometimes there are different types of encryption that may blur the genetic message and make it hardly identifiable. And my scientific interests are in the discovery of unconventional systems in microbes, and specifically unconventional gene expression systems in microbes. And these unconventional systems are found at their best in microbes with complex genome architecture. And I will give you two fantastic examples, which are Ciliates and Diplonema. So I've started working on Ciliates with my postdoc in New York, and I will, as I told you, only give you a brief overview of my research. And I will spend more time on Diplonema, which is the species on which I worked during my PhD. So first, let's start with Ciliates. So in Columbia, in New York, I'm working on Ciliates, and Ciliates are actually huge unicellular eukaryotes that are very far from humans. And I would like to be tall enough to show you things. Still not tall enough. Okay, so Ciliates are here, and humans are here in Metazoa. So Ciliates are very far from humans, and perhaps you know two very famous Ciliates, which are Paramecium and Tetrahymena. And perhaps you know them because in Tetrahymena there were at least two great discoveries that led to Nobel Prize. So the discovery of telomeres and telomerase, but also the discovery of catalytic catalytic RNA. And from a genomic perspective, Ciliates are best known to have two nuclei. So they have a micronucleus. So the micronucleus is the germline genome. It is a huge genome, which is around 500 megabase pair. This genome is diploid, but it's mainly silent, meaning that during the life of the ciliate, there is almost no gene that is expressed, and the genes are only expressed during the mating. And there is also the macronucleus, which contains the somatic genome. So this genome is more compact. It's 70 megabases. It's compact, but it contains a lot of chromosomes. So there are 20,000 small chromosomes that are called nanochromosomes in the MAC genome, and each chromosome is present in multiple copies. So you can have up to 1,000 of copies of each chromosome, and this is the genome which is transcriptionally active. And so is there a relationship between the germline genome and the somatic genome? Well, actually, yes, there is a relationship, and the somatic genome is derived from the germline genome by DNA rearrangement. And actually, during the mating, during the conjugation, there are pieces of this genome that are spliced out, that are stitched together, and then you have addition of telomeres at the end of the chromosome and amplification of each chromosome. So by DNA excision, elimination, unscrambling, so pieces can be scrambled on the germline genome, and then amplification, the macronucleus is built from the germline genome. So this is a fascinating process, actually, but I will not tell you about this process because this is not the focus of my postdoc work. And actually, I told you that I'm interested in gene expression, so we can have a closer look at the genome which is transcriptionally active, which is the macronucleus here. So the macronucleus contains nanochromosomes. Nanochromosomes are of gene size, so they are about 3.2 kb in size, and usually there is a single gene on each chromosome. And on each side of the chromosome you have telomeres, and what you can notice here is that the size of the upstream region, so the region which is upstream each gene, is really small. So this is a problem for gene expression. So how are genes expressed? So where are the promoters? Where are the regulatory elements in these chromosomes? And to add to the confusion, actually, there are also what are called a chromosome isoform in ciliates. So chromosome isoform are just like gene isoform in humans, but at the chromosome, so at the genomic level. So they share some parts, so they are derived from the same pieces of the germline genome. So for example, pieces A and B are shared between the two chromosomes, but the end is not the same. And so you can see that they actually share all their 5' regions, but despite sharing this region, the gene expression is different. So this is another thing which is puzzling. So where are the elements that regulate the gene expression of these genes? And actually, I will leave you with this question because this is exactly the project that I've started in New York, so the identification of regulatory elements in ciliate. So for the moment, I've just started to reassemble this macronucleus genome. And specifically, I used PacBio reads to decipher between the different isoforms that are contained in that genome. And then I will start to search the regulatory elements in ciliates. So this was the first part about ciliates. And now I will switch to diplonemids. Okay, so when I presented like 1 month ago, my work in New York about diplonemids, so my boss introduced me, and so she told the audience that diplonemids are the most bizarre species that she has ever heard of. So coming from someone who is working on microbes with 2 nuclei, I think that this is something. So I don't know if diplonemids are more bizarre than ciliates, but you will see and you will tell me if they are more or not. It's like Batman against Superman. So which one is the most bizarre? So this is a phylogenetic tree, and you can see on this tree a selection of species, and you can see Diplonema, so a diplonemid which is here, and the closest relatives of diplonemids, which are kinetoplastids here with Trypanosoma and Leishmania, and perhaps you know these species because they are well-known parasites of humans. And I show you this tree to draw your attention on the huge evolutionary distances that are between diplonemids and kinetoplastids. And to give you a sense of the scale, you can compare the distance between humans and mice. So the evolutionary distance are much longer. And so these are it has direct consequences on bioinformatic analysis. Because when you take any gene from or any protein from Diplonema and you compare it to any other species, then the similarity will be very low. And this was one of the challenges of my project in PhD. So Diplonema is a very divergent species. And it is a very divergent species, but I must admit also that it's not a very well-known species. And actually, it was not very well-known until recently, where there was a special issue of Science. And this special issue was about published the first results of the Tara expedition. So Tara is actually this schooner, and the Tara expedition is a scientific voyage, and this expedition has different goals, but one of these goals was to quantify the biodiversity in the oceans. And so what they did is that they collected 35,000 samples in the oceans, and then they sequenced the samples. And it did not sequence all the genes, so it's not metagenomic. They sequenced only a small part of the ribosomal RNA, which is the V9 portion of the ribosomal RNA. And with this barcode, they tried to assign the barcode to a branch of a phylogenetic tree. And when they did that, they found that 30% of the species were unassignable. So meaning that 30% of the species that they had are completely unknown or are far too divergent to be assigned to a branch of a phylogenetic tree. So for me, when I look at that publication, I was like, wow. There are so many things that we don't know. So there are so many things that are still available to be discovered. So, and if you look at the portion of the sequence that we know, we have only sequenced a little tiny part of the sequence and of this species. So this is really amazing. So there is still a lot to discover. So a lot of work for us, and I think a lot of amazing discoveries. And I think there is really a huge potential of like unknown system. And it would be interesting to dig into these sequences to find unconventional species, unconventional systems. So still they assigned 100,000 species in the Tara team. And what was quite surprising is that the third species in terms of sequence biodiversity are diplonemids. So diplonemids are the first species in terms of biodiversity in the oceans. And before the work of Tara, nobody knows about that. So it was quite a surprise for everybody. So diplonemids are surprisingly diverse. And diplonemids were known actually before the work of Tara, but it was for another aspect of their biology, and it was their mitochondrial genome. So As you know, so the mitochondria is the powerhouse of the cell, producing ATP, and mitochondria have their own genome. So in humans, this genome is circular and it encodes 37 genes. And these genes are transcribed and translated classically, so almost classically. But in Diplonema, the situation is completely different. So the genome is composed of 100 of chromosomes, small circles, and each chromosome has a short gene pieces, so that are called modules. So for example, there is an example of a gene with 4 modules. All the modules are transcribed in parallel, and then the flanking regions are then trimmed, and the pieces are spliced together in trans, and this is what we call trans-splicing. And finally, the RNA is polyadenylated. And before the beginning of my PhD, so Gertrude and her team has shown that at the junction of the COX-1 gene, there is addition of uridines, so addition of uridines at the RNA level. So this is RNA editing. So this is quite a unique mitochondrial genome structure, and Diplonema has found an eccentric solution for expressing their genes. So during my presentation, I will tell you about trans-splicing and RNA editing. So I would like to recall the definitions. So trans-splicing is the covalent joining of RNA pieces into a single molecule here. And RNA editing is the modification of the coding message, but at the RNA level. And it could be by addition, it could be by deletion or substitution of nucleotides. So what is Diplonema's mitochondrial complete gene catalog? So there are many questions that we could ask on a system like Diplonema, but something that I like to mention, that I always like to mention, is that working with unconventional systems is really difficult. It's really difficult because the tools, so the bioinformatic tools, but also the tools at the bench, so the experimental tools, are not designed for this species. So you always have to adapt the tools. And I will show you in the rest of my talk some examples where the bioinformatic tools just don't work. So you have to find another way of doing things. So even very basic questions like how many genes may sometimes be a difficult question. So first, we wanted to know how many chromosomes there are in the mitochondrial genome of Diplonema. So the majority of mitochondrial chromosomes were identified by Sanger sequencing before the beginning of my PhD. And Gertrude and her team found that there are actually 2 chromosome classes in the genome of Diplonema, so the chromosome of class A and the chromosome of class B of 6 and 7 kb. And these chromosomes are composed of constant region. So you have the shared constant region in black and the class-specific constant region in blue. So the shared are shared among all chromosomes, and the class-specific are shared but only among the chromosome class. The unique part of a chromosome is called a cassette, and the coding part of a cassette is called a module. And so before the beginning of my thesis, so Gertrude has already identified some of the chromosomes, and by DNA sex, so Illumina DNA sequencing, we complete this chromosome set, and we found that Diplonema's mitochondrial genome consists of 81 chromosomes, which is quite a lot. And then we wanted to, of course, identify the mitochondrial genes, and identification of mitochondrial genes is complicated by two facts. The first fact is the low similarity of modules with known mitochondrial gene. So if you just take the sequence of the module and compare it to known mitochondrial genes, so you will find no similarity. And only the most conserved sequences were identified using Markov models. And also, the second point that make identification of genes very difficult is the fragmented structure of genes. And so to find genes, what we decided is to start not from genomes, but from transcript using RNA-seq. So easy. So we just sequence the RNA-seq. And usually what you do when you have your RNA-seq reads is that you take your reads and you map it on the genome. And then, of course, you find the genes. And with spliced mapping, you can find also the introns. So this is a good strategy, but it doesn't work if the exons are on different chromosomes. So this strategy didn't work. Then we thought, well, perhaps we can use tools that are used for people working on cancer cells to detect chimeric genes. So in cancer cell, you have a chromosome rearrangement, so you have part of the genome that are bring together. And so there are tools that could detect these chimeric genes. And this very looks like trans-splicing. And so we use these tools, but the problem is that the tools expect some of the genes to have chimeric junction, but not all the genes to have chimeric junctions. So it didn't work. So finally I thought, well, okay, I will put everything together and just do a de novo reconstruction of transcript, and just to see if something came out of that. But the problem with that strategy is that so transcription in the chromosome are initiated here, so in the constant region and in both directions. So then you have those primary transcript with long flanking region and in both directions. And you have also all these intermediates, so with and without flanking region, edited or not edited, plus, of course, the transcript that you are looking for. And the thing is that there are too many confusing RNA intermediates, plus the bidirectional transcription with long flanking region, so that completely confused the assembler. And so at the end, we obtained quite nothing. So it didn't work either. And so I was like, okay. And so finally, I had to develop my own script. And what I did is that I only mapped the RNA-seq reads on the cassette. And with paired read, I was able to infer the modules that belong to the same genes. And with split reads, I was able to infer adjacent modules. So finally, Diplonema's mitochondrial genome contains 18 genes that are split in 84 pieces. And among these 18 genes, 10 genes are components of the respiratory chain. We have 2 genes that encode for the large and the small ribosomal subunits, so involved in translation. And we have 6 genes which function is completely unknown, so we call them Y, Y1, Y2. And we did not find any tRNAs, but this is not really surprising because tRNAs can be encoded by the nuclear genome and then imported into the mitochondria. So finally, what we can say is that Diplonema, so the structure of the mitochondrial genome of Diplonema is exceptional, but the mitochondrial gene complement is rather classical. But we had a first surprise by looking at the RNA-seq mapping on the genome. And the surprise was that RNA editing by addition of uridines is far more widespread than what was known before. And this is a schematic representation of the mitochondrial chromosome. Each box represents a module, and you can see the position where you have addition of uridines by RNA editing in green. And this process is post-transcriptional, meaning that you can detect pre-edited and edited sequences. And the uridines are always added at the end of the modules. And when they are added at the end of the transcript, they often reconstitute the stop codon. And when they are added between modules, so sometimes they change the reading frame. And there is up to 50 uridines that are added in that gene. So mitochondrial transcript in Diplonema undergo an extensive editing by polyuridylation. And actually, it was not the only surprise that we had while looking at the RNA-seq mapping. And when we look at the mapping, so this is the example of the mapping on the first module of the NAD4 gene. So this is the module. So I guess you are all familiar with this view. So reads, RNA-seq reads that are mapped. So in gray, there is a perfect match, and when there is color, so there are mismatches. And you can see that in the middle of the module, there is a drop in the coverage. So meaning that there are some reads and part of the reads that don't map correctly. And if you look, if we zoom in and you look carefully, perhaps you can see a pattern, actually. And this pattern is the fact that when you have a C at that position on the genomic sequence, you have T's on the RNA. And when you have A on the genomic sequence, you have G on the RNA. And the modification of the coding message, but at the RNA level, is exactly the definition of RNA editing. So what you are looking here is RNA editing by substitution. And what we found in that module is that 29 positions are clustered on a short sequence and are edited in NAD4. Then we were like, wow, this is amazing, so many positions that are edited. And then we wanted to search for other positions that are possibly edited in the genome. So how can we do to detect other positions that are edited? So usually, classically, the approach is that, so you have a reference sequence here, and on this reference sequence, you will map DNA-seq, and you will map also RNA-seq. Then you will filter low quality alignment, you will do a variant calling, and on DNA you will detect DNA variants that are SNPs, and on RNA you will detect RNA variants that are both SNPs and RNA edited position. And by doing simply the difference between these two files, you will have only the RNA edited sites. Okay, so this is good for classical species, but for Diplonemids it didn't work, actually. It didn't work because the reads that cover the edited region have a very low mapping quality. And so usually they are filtered out during either this step or the variant calling. So the mapping quality is too low, so you will not detect the edited sites. And there are strategies that are used by RNA editing tools to detect these positions that are edited. Usually what they do is that they mask the edited sites, or they use a reduced alphabet, so meaning that they encode, for example, A and G by R for purine, or they use a reduced substitution cost. But if you use all this strategy in Diplonemids, so if you mask edited sites, so you have too many mismapped reads, so it doesn't work. And none of these strategies work to detect positions that are edited in Diplonemids. So we started a collaboration with Peter Stadler in Germany, and Peter and his team have developed a tool, a mapping tool, which is called SegML. And in SegML, they have implemented a module for Diplonema. And the turnaround that they found for this specific problem is actually simple but brilliant. And perhaps you know it, but when you map a read onto a genome, there are two steps. The first step is the seeding, and then you can anchor the read at the correct position. And then there is an extension step to extend the alignment. And what Peter did is that his team implemented the fact that you do not allow mismatch during seeding, then the read is anchored at the correct location, but you use a reduced alphabet during the extension, and then you allow mismatches, you allow more mismatches during the extension. And this strategy worked beautifully, actually, and we were able to detect the positions that are edited. And once again, there is an extensive substitution editing. So 29 positions are edited by A to G substitution and 85 by C to U. The edited sites are always clustered, so they are on the same module, and in the module they are on a short region of the sequence. It's also a post-transcriptional process, and the editing rate is high, meaning that if there is a position which is edited, and then it will be edited in 100% of the mature transcript. And we have found, so my colleague at who is working, Matouche, who is working on the experimental part of this project, has identified that A to G is actually a deamination of A to I. So an inosine is read as guanosine by the reverse transcriptase. And this is the first report of A to I editing in mitochondria. So finally, we have found that there is an extensive clustered RNA editing by A to I and C to U deamination in Diplonema. And just to recap, I will give you the example of RNA editing in NAD4. In NAD4, we have two types of RNA editing going on. So RNA editing by U appendage between module 4 and module 5. So module 4 and 5 are transcribed. Then the flanking region are trimmed, are clipped. There is addition of uridines at the end of the module 4, and then splicing in trans between the two modules. And you can see that because there is addition of two uridines, then it causes a shift in the reading frame. And there is also substitution editing in the first module. So you can see the pre-edited sequence and the edited sequence here. And you can see with the translation that RNA editing renders the protein more hydrophobic. And this is actually something which is important. If you know that NAD4 is a component of the complex 1 of the respiratory chain, and actually NAD4 is a transmembrane protein. And the region which is edited is this region in blue. And if you and you understand that if you have polar amino acid in that region, then that region will be disorganized. So actually RNA editing in Diplonema correct the message. And this is something which is important. So editing is important for function integrity in Diplonema. And this is not always the case. So, I mean, there are other cases of RNA editing which is known in other species. And I will give you 3 examples. So, RNA editing is known in humans, for example, and there is a very famous example with the ApoB gene. And the ApoB gene, so in the liver, the protein which is expressed corresponds to the gene which is encoded by the genome. So there is no difference. But in the intestine, there is RNA editing of a CAA codon that is changed UAA. So it's a C to U modification. And UAA is a stop codon. So in the intestine, you have a shorter protein. And this is very interesting because in that case, you don't have a correction of the function, but you have a diversification of function. And in humans, there are also A to I deamination, but those events occur in allele repeats and non-coding sequences like introns or 3' UTR. And there is also RNA editing in plants. So in plants, it's usually in the mitochondria or in the chloroplast. So I forgot to mention, but in humans, it's in the nuclear genome. So in plants, it's in the mitochondria or the chloroplast. Usually, it's a C to U or U to C RNA editing, and you can see with this screenshot that the positions that are edited are scattered and not clustered on a short region. So with 3 types of editing, C to U, A to G, and polyuridylation, and given the number of edited sites in Diplonemids and the cluster distribution of the positions that are edited, so that make RNA editing in Diplonema quite exceptional. So another question that we could ask with that system is what kind of machinery performs RNA editing and trans-splicing? So what are the proteins that perform this process? And before answering to that question, I will tell you what kind of machinery does not perform RNA editing and trans-splicing in Diplonemids. Because actually we know machinery that perform trans-splicing. And this is related to the introns. And actually, we know 4 types of introns. We know the spliceosomal introns, which are the classical introns, the group 1 and the group 2 introns, and the archaeal tRNA introns. And in these 4 groups of introns, we know cases of trans-splicing. So meaning that if you have splicing, you have trans-splicing. And so we search for signals that are typical of these introns in Diplonemids, and we did not find any. And of course, we search also for complementarity of flanking regions, flanking regions for contiguous modules, and there is no complementarity, no obvious complementarity. So trans-splicing in Diplonema mitochondria is different from all known mechanisms, and it is not specified by complementary cis signals. So this is a new types of introns, actually. Is the trans-splicing or editing machinery guided by trans-RNA-factors? So this is also a very popular hypothesis. So the hypothesis is that perhaps there are antisense RNA that could guide the modules that are contiguous, or perhaps there are antisense RNA that could specify the positions to be edited. And this hypothesis comes actually from the sister group of Diplonemids who are kinetoplastids. And these species have also a very strange mitochondrial genome. They have a genome which is composed of a maxi circle which encode genes and mini circles that encode guide RNA. And the genes are transcribed and also the guide RNA, and they are positioned that needs to be edited by addition of uridines or deletion of uridines on the mRNA. And the guide RNA are antisense RNA that specify the sequence to be edited. So the hypothesis was, well, perhaps it's the same thing in Diplonemids. And so we search in a library of short RNA-seq, so stranded short RNA-seq. We search for guide RNA, and we did not find any. So guide RNA are probably not involved in trans-splicing and editing. So then, what kind of machinery performs RNA editing and trans-splicing in Diplonema's mitochondria? Well, we don't know yet, but we have some hypotheses, and I'm running out of time. So we search for candidate function in the nuclear genome. So we sequence the nuclear genome, we assemble the genome, we annotated the genome. So this is only one sentence, but it takes time. And we search for candidate function, and we found actually some interesting function. So in conclusion, I told you that oceans contain an amazing diversity that could reveal fascinating features, that diplonemids are surprisingly diverse. I told you also that mitochondrial genes are decrypted by RNA editing, by polyuridylation, RNA editing by substitution, and trans-splicing. And I show you also another interesting system, which are ciliates. So ciliates are microbes with 2 nuclei. And in the macronucleus of ciliates, which is the genome, which contains the genome which is transcriptionally active, the expression of the somatic genome is essentially unknown. So with that, I would like to thank the people in Montreal. So the team is led by Gertrude Berger. So I would like to thank all the people in the team, and also our collaborators in Germany, and people also in Columbia University. So perhaps you know Laura Landwerber, who leads the team. She was in Princeton so last year, and now she moved to Columbia. So yeah, so this is it. So thank you.
That's a lot of material, so we'll just take one very quick question, and we can continue the discussion over snacks. Yes.
How do the diplonemids survive if it seems like so much is required for them to maintain homeostasis? And it can't happen randomly. There must be like a very organized way to make all of these editing very efficient process.
Yeah, but it doesn't seem to be detrimental for the survival of the species, because I mean it's a really popular species. So I think that the species found a way to make it more efficient. And actually, if you look, if you take all the RNA-se reads and you look at what is going on, you see that actually all the processes are occurring in parallel. So I think that it's more or less like in computer. So it's parallelization of process that makes things perhaps highly efficient. And so actually it works very well. And if you look at mistakes in the RNA-se reads, you can find almost no mistakes. So it's really very efficient, and so efficient enough.
Great, thanks.